our next thing after general senses is special senses. These are ones where the senses are located in specific parts of the body and do a very specific task. So they're not going to be located throughout everywhere, just one spot or two spots usually. Things like olfaction. Olfaction is the scientific name for the sense of smell, the ability to take in odors, or chemical sensation of that nature. Chemoreceptors located inside the nasal passages have little small hairs attached to them. As the hairs trap the little chemicals, they funnel them down. The right shape signals the right signal to be sent forth. So as you breathe in, these chemicals come in, they hit those hairs, they go up along there. So that is why when you try, if a nose hair gets pulled, it's really painful because it's so closely connected to a nervous system, and therefore it is not like the normal hairs here, which have nerves there, but they're not like really heavy nerves. In this case, it's going to be a problem. It's going to be trying to stop you from damaging those ones, which are trapping all of those senses in your nose and funneling them down to these cells. So the olfactory hairs trap them, push them down there, the right shape. Now, does everybody smell the same things? No. Everybody has different receptors. You have general receptors that most people have, but some people can smell certain smells and some people can't smell the smells because they have the different shapes of the different receptors. So that maybe you have the wrong one. That your shape is a square and the chemical that you can't smell is a circle. Someone tries to go in, it won't work. Same kind of concept there. The olfactory cells are the ones that do that task. Olfactory bulbs are the actual portions off the bottom of the brain, which are two small portions of the olfactory nerve, which receive all the information from all these cells as they come in here and then transmit it up the olfactory nerve into the brain so you can get your sense of smell. Gustation is the sense of taste, closely related to the sense of smell because they're both chemoreceptors and they both have an overlapping thing. And some animals do the same thing, things like snakes. So the taste buds are these chemical receptors. There's about 10,000 in an average mouth. Are there people with more? Absolutely. They call them super tasters. They have more than the average person, which means they can detect small differences that you and I can't. They taste a piece, you and I taste a piece of food, taste a certain way. They taste it, they can tell all the different individual elements to it, the different individual parts of the food, because they have the ability to taste each of those separately. So the papillae are the bumps that are found on the tongue. They contain the taste buds. Can you grow back taste buds? Absolutely. They get damaged to one of the few parts of the nervous system which can grow very easily back. But if you damage them too much, they can become scarred and not work. There are four primary tastes: sweet, sour, bitter, and salty. Now, these are all located to things like sugars, acids, bases, and salts. They detect those basic chemicals, so are able to allow us to do those kinds of concepts. Are they located in specific spots of the tongue? No. They're actually located throughout the whole tongue. It's just some spots have higher levels of one than the other, but they're not located just in one spot, and that's it. It doesn't make sense from an evolutionary standpoint to be protective if you can't have these, because if you can only taste sour in one portion of the tongue, if you eat a food that gets past there, it has a lot of heavy acid in it, you'd be hurt before you'd know it. So it has to be in all the tongue that way. The other one, which is a fifth sense, is umami, which is savory, which is found in a lot of Asian food. We get that from those kinds of foods. Vision is our next sense. It's our number one sense when we come to our general senses, obviously located in our eyes. It is our most complicated sense. It has the most things involved with it. Our brain spends a lot of its energy involved in this particular sense. It has a lot of structures that go along with it, such as eyelids and eyelashes. They both have protective functions. Their job is to protect things coming into the eye from damaging the eye and the sensitive nature of it so that you can actually continue to do these sensations. The lacrimal gland, otherwise known as the tear glands, their job is to produce fluid, which is antibacterial, which is also to try to sweep out any sort of particles that get in there and to keep the cells from drying out. The muscles of the eye, there are a lot of them. They're all tiny little small muscles that do multiple little intricate small motions. So when you have a camera and you're trying to adjust and sort of focus in, these guys do that by changing the eye around and moving it and helping to push it to where it needs to go. The structure of the eye, how it's set up. It has layers we call tunics. The first one's called the fibrous tunic, which is made up, as the name implies, fibers of proteins. The first part is called the sclera. You know the sclera as the whites of your eyes. This is the part that you see. It actually goes all the way around the outside and helps protect it. It's nice and sturdy and durable. Is it indestructible? No, you can damage it. People do get it punctured by things, so you gotta be careful with it and don't go poking it all the time or scratching it. The cornea is the clear, transparent front window. It's the part we see here in the front that sort of bulges outward. Over time, it builds up layers which can damage its ability to actually focus. So you may have to wear things like I do. Or you can have problems with the lens behind like I do as well, which causes those things. People have all kinds of issues with both of those things. So the cornea can be transplanted, it can be replaced from, one person can donate them after they die, and it can be transplanted to another person if you have problems with it. But it's going to keep everything inside there from draining out from the center of the eye, which is where there's an opening there, so if you don't have a window, you would have it all drain out. So here you can see sclera's on the outside, here's your cornea, and over time you will build up layers to make it stiffer and harder for it to focus, for it to do its job of being flexible and helping to adjust with the eye. The vascular tunic, as its name implies, is full of blood vessels. This is the layer full of all the blood vessels, which help us to keep the eye alive and functioning correctly. You can see these sometimes through the sclera because they become large enough that you can see them become bloodshot. The choroid membrane is a brown membrane that absorbs light, because if you didn't absorb extra light, it's like having a camera with it just basically wide open and have a bunch of light there. It'll just make this, it'll wash out your image. You won't have a good image. So for your eye to see, you have this brown membrane which absorbs a lot of that. Does it absorb all of it? No, if you put too much light in it, it's going to be overloaded still and you can't see. The ciliary body is a small, smooth muscle which connected to your lens helps to adjust and pull the lens in order to help you focus in on an object better. The iris, just like the iris in your camera, is actually opening and closing to allow light to come in. It's the colored portion of your eyes. Its actual purpose is it will open and close the small hole in the center of it called the pupil, because that hole is an actual opening. If the cornea wasn't there, yes, the stuff would leak out, because that is actually a hole there. It's not just like a magic brown spot. It's actually a hole that goes into your eye that allows light to pass in, just like there's a hole in the camera that allows light to come in for the images. The lens focuses object. It's a protein-based one. It over time also builds up layers, can make it become stiff and make it have ability as you get older to have it harder to see because of that reason. So its job is, just like any lens, it's going to invert the image. So the image comes in, it's actually going to flip it upside down and backwards as it goes in the back, and your brain's going to flip it back the right way. Cameras do this by having multiple lenses. Our brain does it by having the brain be able to just figure out that the image that looks like this should actually be like this. Inside here is also what's called the aqueous humor. It's a water-based liquid that helps give it the shape of the eye. But if you just have like a bag full of water, if you have a water balloon, you know what happens if you lay it down. It just squishes down. So in our eyes, if you just had water there, it only just goes to the bottom and forms sort of a flat layer. I need a little something else there to have to give it a little bit more shape. That's where the vitreous humor comes in. That is a gel-like fluid along with the water that helps it to keep it from collapsing inward. Does it mean it can't do that? No. If you lose some of the stuff, like you see people have problems with their eyes over time, they may actually have an issue where it starts to deflate and basically the eye starts to shrink down in size and sort of fall in like a water balloon that's been sitting down too long. So in here, here's my ciliary body, choroid membrane's up here, iris is around the outside, the pupil's the hole here, here's my lens. Inside here is where I'm going to have my vitreous and aqueous humor. The nervous tunic is the last portion. This is the portion that's the nerve tissue, where it's picking up the information. It's got all my photoreceptors. Photoreceptors are located in a region called the retina, which is the back of your eye. It looks kind of like a movie screen, like an old school movie screen. It has like a bluish, sort of silver appearance, kind of shiny and bright colored. Inside of it are two basic types of cells, rod cells and cone cells. They are the types of cells that pick up different information. They're photoreceptors. The rod cells work in low light, so you don't need much. They work well at night. So things like no color, black and white. So you can see things in very good clarity in low light, but you don't have all the different contrasts of picking up the light to see the colors. They work on a chemical called rhodopsin. Rhodopsin needs vitamin A to work. It's one of the reasons why people always thought that you need to eat carrots to make your eyes better at night. They don't make them better at night, just don't make them worse, because vitamin A is found in carrots. So it's actually something that's useful if you eat certain types of vegetables that are going to give you plenty of vitamin A. They keep you from having problems. If you don't have enough vitamin A in your diet, you get what's called night blindness, which means you have problems seeing in the night, in the dark, because you can't even use these cells to work when they should. Cone cells are the exact opposite. They work because they're color sensitive. they work really well with lots of light. If you don't have enough light, they won't work very well. They detect 3 basic light color patterns. Now colors are basically just wavelengths of light. They detect the wavelength of red, green, and blue. That's the only 3 they detect. You only have those 3. So your brain takes those 3 and determines intensity levels of those and overlapping to create all the different shades of color that you see. So your brain is able to mix them to create that palette. So you can see here, the rods have this nice curve here where they detect light there. Blue cones detect here, green cones detect here, red cones detect here, and that's how they detect your light. And they mix these overlapping patterns at different intensity levels to create different shades of color. When you see these, you can see, here are my rods, here are my cones. They're hard to tell. It looks a little easier here to see the rod and the cones when you see them under a micrograph this way, to see what they look like. And that's it.